These are your video notes for Hardy-Weinberg equilibrium, how to determine if populations are evolving, if they are changing over time. First, some vocab. You've seen this before, but good to go over it. Remember that the genetic makeup of an individual is its genotype. You can be homozygous dominant, heterozygous, or homozygous recessive for any trait or any gene. A population consists of many different genotypes, right? And therefore, a population consists of many different alleles for those genes, because you have some dominant alleles and some recessive alleles. So all of the alleles for a gene, for a trait, that exists in a population is referred to as the gene pool. And we can calculate the percentage of those alleles. How many of the alleles in the population are dominant? How many are recessive for any given trait? And that's what Hardy-Weinberg does. If these alleles change frequency over time, that's evolution. Evolution has happened. So if we can quantify this, we can figure out if evolution happens and how quickly in populations. Remember that evolution is just change over time. It's change of allele frequencies over time. And that's influenced mostly by natural selection, but there are other factors. So there are 5 factors that I'm going to talk about here with you now that make populations or contribute to them changing over time. Natural selection is one of them. Being able to choose your mates is called sexual selection. Right? You pick your mate. It's not random. For some species it is, but for humans and a lot of other mammals it's not. For some animals it is, but not mammals. Mutations, changes in the genetic code, drive evolution. Migration, new individuals coming into an area, bringing in new alleles, or individuals leaving and taking their alleles with them. Right? And finally, random events. It's cut off here, but this is usually most significant in small populations. This is referred to as genetic drift. Genetic drift. And these are events that change allele frequencies regardless of natural selection or anything else, called genetic drift. So let's go ahead and summarize. So here, just really quick, light causes what are called heterodimers to form. Basically, you don't need to know that, but that changes DNA, causes a mutation. As individuals migrate and move, they bring their alleles with them in or out. Sexual selection, choosing your mates. This is genetic drift, so an event that changes the allele frequencies, not because there's a benefit to being a red bird on this island over here out in the ocean, but just because those are the birds that showed up. It's kind of like random chance, which is different than natural selection, which is not random chance, but genetic drift is. So there are elements of chance in here, but natural selection is not part of that. That's genetic drift. And then natural selection, right, which is very much not random. So those are your 5. And here's where we get into it. Allele frequencies in populations do not change, which is to say there is no evolution if, and all of these criteria must be met. There must be no natural selection. So all individuals have an equal chance of reproducing and passing their genes on to the next generation. Mating must be random. Individuals don't choose their mates. There must be no new mutations. No new alleles are being created. There must be no migration in or out. And the populations must be very, very large. And if you're looking at this set of criteria and you're like, this is impossible to meet, there's no way in nature all 5 of these criteria are true. And you would be absolutely correct. Hardy-Weinberg equilibrium the equilibrium, the set of conditions that results if these 5 things are met, is not what actually happens in populations. But that actually is useful for us because depending on how much of this is not true, depending on how much natural selection or random, non-random mating or et cetera there is, lets us compare it to a baseline. So basically, this is a baseline. This is the criteria, the 5 criteria points that establish a baseline. And based on the math, which we'll do in a second, you can figure out there's almost always going to be evolution. It's an unavoidable fact of life, you'll find out. But how quickly, how significant, and that's where the math comes into play. So here's Hardy-Weinberg. Again, it is a tool to help us figure out if a population is evolving or not. And again, realistically, the answer is always yes. Meeting those 5 criteria in a population is virtually impossible. So yes, things are always going to be evolving. That's the point of the unit. So yeah, if all 5, I was just kind of deciding how I wanted to paraphrase this. If all 5 of these things are met, then yes, there should not be any change. But the whole point is that at least one of those criteria will inevitably be violated, right? And you will have changing allele frequencies. It's impossible to not have changing allele frequencies. You're always going to have mutations or individuals leaving or random events or natural selection. You're always going to have at least one of those. You're always going to have evolution. So none of these assumptions are going to be true all of the time. You're always going to have violations, and therefore you're always going to have evolution and change over time. So let's jump into some math. We're going to give all of these alleles letters, and the notation is what it is. I can't change it. I don't remember who came up with this, but there's actually an error here. This should be the letter Q. So the letter P, lowercase p, is the frequency of the dominant allele. So for us, maybe it's capital A. The letter Q is the frequency of the recessive allele, so the lowercase a. And if you add up all those frequencies, they always equal 1. So let's say if half of your alleles are dominant, P is 0.5. If half of your alleles are recessive, Q is 0.5. You add up all of your alleles and you get 100% or 1. And using decimals will be your best friend. You're not really going to want to use percentages or fractions really at all. You're going to want to stick to decimals. For the genotypes, so that's one equation. For the genotypes, P squared is the frequency of homozygous dominant individuals. 2PQ is the frequency of the heterozygous individuals. And Q squared is the frequency of homozygous recessive individuals. And if you add all of these up, all your homozygous dominant, homozygous recessive, heterozygous individuals, you should get all of your individuals, 100%. So if you add P squared, 2PQ, and Q squared, you get 1. This is your second equation. So let's walk through problem-solving hints. If you know the frequency of one allele, either the dominant or the recessive, you can figure out the frequency of the other. Remember that P plus Q equals 1. If you know the frequency of the homozygous recessive individuals, the actual genotype, the lowercase a, if you know Q squared is what I'm trying to say, you can figure out Q by square rooting it. And then once you know Q, you can figure out P. And I'll show you here in a second. Always use decimals. And remember that the first thing you're going to do in these problems is figure out what piece of information you are given. What are you given, and then what can you solve for? Also remember that the frequency of the dominant phenotypes consists of both P squared individuals, homozygous dominant, and heterozygous individuals. Don't forget that the heterozygous individuals show up as the dominant phenotype. So here we go. In a plant population, red flowers are dominant to white flowers. If you have 500 individuals, so in a population of 500 individuals, 36% of the plants have white flowers. They want us to calculate the frequency of all the genotypes. How many individuals would you expect to be homozygous dominant and heterozygous? Right? So what are we going to start with? Well, they give us 36% of the plants are white. Is that P? Is it Q? Is it P squared? Is it 2PQ? Or is it Q squared? It's Q squared, right? If you have white flowers, you are homozygous recessive. So 36% of our plants, 0.36, are homozygous recessive, and that's the value for Q squared. What do you want to do next? Well, I mean, you can just go ahead and read it. The key is right here. Since you know Q squared, you can square root it and solve for Q. So Q equals 0.6. All right, so we started with Q squared. We've solved for Q. Well, now you can plug and chug. You know P plus Q equals 1, so P must be 0.4. All right, now we can answer A. How many individuals are homozygous dominant? Capital A, capital A. Well, the frequency of the dominant allele is 0.4. And if you have two of those alleles, you're going to multiply those together. We're asking for P squared, right? So 0.4 squared, 0.4 times 0.4 is 0.16. 16% of your individuals are homozygous dominant. Plug and chug for your heterozygous individuals. 2 times P times Q gets you 0.48. 48% are heterozygous. And if you want to check your work, and you do 0.6 times 0.6, Q squared, you'll get the 0.36, which we actually gave you at the start of the problem, right? So that's A. How many individuals would you expect to be homozygous dominant? Well, there's 500 of them, so 16% of 500, 0.16 times 500, is 80 individuals. And same thing for heterozygous, 0.48, 48% of 500 individuals is 240. Right? Remember, this is only true if all 5 conditions in this population for Hardy-Weinberg are met. And the whole point is that if you did this exact same problem with this exact same population with 500 flowers maybe 10 years later, if you did this later, you would expect to see different allele frequencies because something will have happened. Natural selection will have happened, mutations will have happened, something will have violated one of those 5, at least one of those 5 criteria. And if you did this in the future, then you would totally expect these allele frequencies to change and you'd have evolution. And that's why we can use Hardy-Weinberg to figure this out. So we'll work more on this in class. I hope this helped, and I will see you then.